Okay, so we just finished talking about the 4 different processes that are that are involved in passive transport, the movement of materials without the cell using energy to get the material in. And, they were simple diffusion, facilitated diffusion, osmosis, and filtration. Now, when we talk about active transport, we had said this, and this we hearkening back to those transport proteins. We had said that this is a process that requires energy. And in general, this is going to be, so if we remember, what was diffusion? Diffusion was driven by this vibration, Brownian motion, and the bumping of molecules into each other. Here, that's not what's going on. In active transport, it is the movement of molecules from an area of low concentration to an area of high concentration, the opposite of diffusion. As a result of that, you have to use energy to make it happen. It's not going to happen on its own. It's going to go the other direction. So this is going to require the cell to do work. What does that mean? Use energy. So we have these transport molecules, and transport proteins, and we've already said that they are, very specific, and you only have a certain number of them, and you can exceed their capacity. That's the 3 things we say about these transport molecules. And so they pull something particular in. We had said you have the sodium-potassium pump that's pumping sodium ions out at the same time as it's pulling potassium ions in. And then we have a great need for glucose in the cell. So you're all the time reaching out and grabbing one and saying, "You're mine. I need you for energy." And pull you in, right? Okay, so that is sort of molecular level active transport. But we also said, we're back to the amoeba. We always talk about amoebas, white blood cells. So if you don't like to think about amoebas, think about white blood cells. We're back to the white blood cells. What did we say they do? They're crawling around looking for something that isn't you, non-self. And what do? They eat it. This is a process we called phagocytosis, cell eating. And in cell eating, it requires energy. So this is an active process as well. Active transport in general are transport processes that require energy. Now, before we were talking molecular level processes. Here we're talking particle level processes. So we can think of this as bulk transport, bringing bigger stuff in. But it is an active process. Cells can bring in a solid, that's phagocytosis, or they can bring in a liquid, that is pinocytosis. Hopefully you're playing along. You see it right there on the slide, right? Okay, so we said, what has happened? You have this particle here in the middle or a fluid, and it gets surrounded and pulled in as a vesicle. We have something else. It's called receptor-mediated endocytosis. So phagocytosis is a self-feeding. A more generic term for bringing bulk in would be endocytosis. It's the same thing, coming in. So what if we think about, we had said at the beginning, again, going back to those proteins in the cell membrane, we had said we have receptors. And we'd said on that cell membrane, we have this receptor, and when the hormone binds to the outside of that receptor on the inside of the cell, that receptor changes shape and causes a change in physiology. It causes your cell to do something differently. Now, if we think of hormones and, changes that happen on a monthly, or a periodic kind of a scale, you say, well, you want the hormone to cause the change, but it's not forever. It should have a limited duration. Don't worry, it does. And the reason for that is because of the next slide. Look at your slides. The next slide is receptor-mediated endocytosis. Remember, endocytosis, bringing something in. Receptor-mediated, caused. So when the receptor has a hormone attached, it gets pulled in. And so you still have that protein sticking out there causing the change, but what did we say happens with a lot of stuff that gets brought in? Phagocytosis. We'd said, oh, you got that lysosome that's going to go over to it, combine with it, and eat it up, digest it. That's what happens. So we have a hormone will attach on the surface of the cell membrane. It gets pulled in, and as a result of that, it's causing a change inside of the cell. And then it gets pulled in, and so that the change isn't permanent, it's a temporary change. You got this lysosome that's going to come over, combine with it, and digest it. And that limits the length of time that hormone binding to the receptor will change the physiology. So what about, we had said that in addition to phagocytosis, cell eating, pinocytosis, cell drinking, or receptor-mediated endocytosis, in all cases you got some waste, right? You did this digestion thing, you have some waste. Well, you can go the other direction too. You can stick it outside, and that's called exocytosis, putting it to the outside. So after the white blood cell digests something or drinks something, or you have this receptor-mediated endocytosis, the waste goes over to the cell membrane, and it gets released to the outside. What about AIDS? So we have AIDS, and it involves, so when we talk about AIDS, how do you catch it? Well, sex, right? It's transmitted sexually, and yet what does it infect? Blood cells. So it has to get, from there into the blood cell. So what happens is we have something called transcytosis. So transcytosis is a combination of endocytosis. So you're in one location and you pass into the cells that are lining that location, and then out the other side into the other location. So if you're in the vagina and you need to get into the capillaries, you go through those cells through this process called transcytosis. The viral particles do this. They go endocytosis in, exocytosis out, and then on their way, they into the capillaries and eventually into the blood cells that they infect. Okay, 9 minutes. We're going to change gears. We're going to talk now about cancer, everyone's favorite topic. All right.